Up next, we have Dr. Ken Yamada. Dr. Yamada is a distinguished NIGCR intramural investigator. His research focuses on the dynamics and mechanisms of cell-cell and cell matrix interactions during cell migration and embryonic development. He has hundreds of publications and citations on topics ranging from salivary gland development to cell adhesion, migration, and cancer invasion. Today, his talk will be on RNA sequence analysis of murine salivary gland development and how that relates to data stewardship and sharing. Thank you, Ken.
Okay, so I'll be turning to a different area, which is opportunities for data sharing in terms of salivary gland development. And this particular field, if you just search PubMed, you can see under salivary gland development, there's over 3,000 papers, some of them, of course, having to do with salivary gland cancers. But basically, there's a lot of researchers in the field who I think could join in to FaceBase. So, as we all know, two of the largest glands are the submandibular and the parotid, and they're different in terms of their gene expression patterns. So submandibular gland, of course, is mixed serous, but also particularly mucus secretion and unstimulated saliva. Parotid gland actually focuses on serous secretion without mucus and stimulated saliva. So these differences, in fact, show up in terms of transcription. So some years ago, Jim Melvin's lab compared adult mouse submandibular and parotid glands and found that, not surprisingly, gene expression patterns were quite different for submandibular and parotid glands. That was in adults. So we and a number of others in our field have focused on budding and branching morphogenesis, which is a process when we, in our field of development, substantially increased epithelial surface area to produce enough saliva. So this is a complex process. Here's a movie showing what you can see in 3D organ culture. So you start out with an epithelium. It's surrounded by connective tissue mesenchyme, and what you have is an iterative process where the gland is divided up here in 3 parts, and then that just repeats with each of the subdivisions, 2 or 3 divisions of the gland until you have a complex branch structure. So this system has proven useful for trying to understand the mechanisms of development and actually with relevance to various other organs. But also it's an opportunity to try to change cell fate or differentiation between the 2 types of glands. And the idea, of course, is to try to apply this to tissue engineering and organ regeneration or replacement. As you know, in Sjogren's or after radiation-induced xerostomia, it's a major problem after head and neck cancer in terms of salivary gland function. So Ray Sakaguchi in our lab and Daniel Martin in the genomics core, who should actually be giving this talk, examined expression at an early stage of development at single cell resolution. And what you can see is in this TSNE analysis that, in fact, there are obviously differences between the 2 glands, even at the single bud stage. So if you look, for example, in magenta, the mandibular mesenchyme, it's quite different in terms of the pattern of gene expression. Of course, each dot is a single cell in terms of expression from parotid and mesenchyme. And then if you look at the epithelia, submandibular epithelium in cyan is different from parotid epithelium, but there's a little bit of overlap. Okay, so they're different. Now, if you actually look at what tissues are involved, one surprise is muscle-related gene expression. And so if you actually look on the right here at the parotid gland, just 3 days later, you can see in yellow here, ACTA2, so it's a muscle or myoepithelial-related gene. And it turns out that in the parotid, you have this development of muscle that we think is involved in squeezing saliva out, whereas in fact, you can actually see striations in that muscle precursor. In the submandibular gland, though, it's quite different. So if you look at the arrowheads, you see this yellow staining on the tips of the buds, and these are actually precursors for myoepithelial cells. And yet, if you look at the parotid gland, you really do not see that type of staining. So, and that will become important later. Okay, so just to summarize at the single bud stage, there's already major differences here at a transcriptional level. Submandibular gland is a simple epithelial bud. The parotid gland, though, has some neuronal fibers as well as these muscle precursors. And that was published a while ago. Okay, so these then allow the potential for hopefully re-engineering gland cell fate by tissue recombination. And so again, Ray and Danny were involved in this, where if you start out with the submandibular epithelium and submandibular mesenchyme, and you have a parotid epithelium and parotid mesenchyme, the idea is to try to swap them. Okay, in other words, you put the epithelium from submandibular with parotid mesenchyme and vice versa, and to see if there's plasticity, if you can change cell fate. So if you look at, in this case, it's a UMAP analysis at parotid in magenta compared to submandibular in cyan, again, you see differences. Actually, on the bottom left is epithelium. They're different. And on the more to the top and to the right is epithelium. Mesenchyme. Okay, what happens if you swap? You see changes. In other words, if you look at the yellow or orange, you see changes in gene expression here. And now, these data are currently in press. They've been deposited in FaceBase, and I'll explain later that there are a number of opportunities available for using that data, of course, using fair criteria. And then if we look at the other exchange, that is submandibular versus recombining submandibular mesenchyme with parotid, epithelium with parotid mesenchyme, again, we see differences in gene expression. So we're able to change the gene expression patterns by swapping with tissue interactions. So what are the specific genes? So you can do so-called volcano analysis, where more towards the left or to the right, or decreased or increased expression of specific genes in the epithelium or in the mesenchyme, and you can see differences. And you can then look at count the number of differential expressions of different genes. And so on the bottom, in submandibular epithelium, closer with parotid mesenchyme, you can see over 600 changed gene expressions, and on the right, also a number of changes with parotid epithelium, closer with submandibular mesenchyme. So what's changing? Well, one interesting change is if you, again, we go back to this ACTA2, which is a marker for myoepithelial cells. In the submandibular gland, you can see it present, it's yellow. And in the parotid gland, you don't see it, of course. But when we swap and look at the parotid epithelium confronted with submandibular mesenchyme, we've switched the fate of these cells now to more myoepithelial fate. However, if you look at submandibular glands and you put them with parotid mesenchyme, they are still committed to forming that. So there's this partial flexibility or plasticity in terms of fate. In addition, parotid put with submandibular mesenchyme starts to express mucus genes like MUC19. You can do further analyses, and I won't go into this, but for example, by bioinformatics, you can predict various ligand receptor interactions that are perhaps upstream of these changes in expressed genes and identifying possible missing molecules. So that's been focusing on expression, but I'd like to briefly consider the cells themselves. So this is work of Xiaohe Wang, looking again, in this case, in submandibular gland. As you look at the green nuclei of these cells over time, and you can look at actually almost every cell in a developing gland, you can see a lot of movement, which is somewhat puzzling, but you can also see some order towards the outer boundary of the organ. You can then track individual cells to see their speed and where they're going. And then you can try and compare that first to gene expression. Now, one other critical experiment is, these cells, if they're individual cells, can they actually reassemble a primitive gland in terms of morphogenesis? So the idea is you dissociate the cells, the single cells, and then over 2 days, they are able to reconstitute a budding gland with ducts. So this tells you then that single cells can actually undergo this process, which then raises the possibility of then applying data from gene expression to actually look at this reconstitution process. So if you look further into single-cell RNA sequencing, you can have different clusters that indicate different gene expression patterns in the outer bud, or inner bud, outer duct, and so forth. And you can then do so-called RNA velocity analysis. That is looking at spliced and unspliced mRNAs to try and predict where these patterns will go. So, for example, you see this swirling pattern that seems to be going towards the outer duct of the outer bud cells. So that was interesting. And what's going on in the swirling pattern seems to be just different phases of the cell cycle. Okay, then you can take these types of data and look at expression of different genes. And I'm not going to go into this. We have not looked into these in detail. That's why we're sharing the data. But we focused on 2 types of genes involved in cell adhesion. So here, then, you see in this analysis increased expression of integrin receptors for matrix proteins, particularly basal membrane. And then here's another gene that's decreased strongly. This is the cell adhesion protein, cadherin. Okay, so just taking these 2 genes, you can actually then begin to understand some of the processes involved. So looking at the cell-to-cell adhesion molecule, cadherin, so the green indicates its localization in the developing gland. So the inner bud has quite a bit of staining, but as you go towards the outside, to the outer bud cells, near the yellow basal membrane, very puzzlingly, you see a decrease in the levels of expression. So the idea then is to try and take this information, that is reduced cell adhesion molecule, cadherin, increased interactions or increased expression of integrins and try to re-engineer this process with a cell type that actually never goes through this budding process. This happens to be the human adenocarcinoma line that just forms spheroids in culture. And Shahu was joined by Kazuhiro Matsumoto. And bottom line, this has been published, is that if you experimentally downregulate E-cadherin using CRISPR-based approach, and then provide integrin targets in a basal membrane, you can actually reproduce this process. So, for example, here on the left, you can see these cancer cells, the spheroids actually undergoing budding. And then on the right, it's a somewhat later step, very active. And actually, if you focus in on that, you can see very active cell movements, but also it's organized, where in fact the cells that are magenta, that have low E-cadherin expression, are right out there near the basal membrane and starting to form this bud. Okay, so just in conclusions, RNA-seq analysis, particularly single-cell sequencing, is generating a lot of raw data in terms of gene expression, but also in terms of imaging. And we're hoping that some of the imaging data can also be imported into FaceBase. We have hundreds of differentially expressed genes that have changed, and where obviously a wide variety of bioinformatic analyses are possible. But we've only focused on a few of these genes, so we hope others then will work on some of the other genes that might yield some other new insights with many opportunities. And for many of us who are struggling with this NIH data management and sharing responsibility, I think FaceBase is a really great way to do that. So thank you.
Any questions for Ken?
Ken, beautiful talk. I was just wondering, when you did epithelial mesenchyme sort of recombination study between the submandibular versus the parotid, seemed like the epithelial has the instructive signal for the branching part, right? And is that stage dependent, meaning that if you do it at a later stage, would that change?
We really don't know about a later stage because it's technically difficult. Because if you have all these buds and so forth, try to separate out the mesenchyme from the epithelium, it's very difficult. So we don't know that. I didn't mention though that in fact the epithelium can also, conversely, besides mesenchyme instructing epithelium, instruct mesenchyme. And so parotid epithelium can actually induce the submandibular mesenchyme to start producing muscle-associated proteins. So, it's a two-way street in terms of communication.
I have a question, just in terms of the data volumes I was going to ask, in terms of the data volumes and data types that you have videos. How many of these do you have? How do you do any sort of analysis based on fluorescence imaging? And in terms of trying to store this data, how have you been storing them now, and what's your plan for it later on?
So we obviously we have quite a bit of data. We have multiple terabytes of data in terms of imaging. I can tell you that a FaceBase will be great because we really did try to fulfill NIH requirements in terms of data sharing. So we put some of our data on Figshare, the older data. And it cost us just under $10,000 for one paper, and because of all the imaging data. So we're hoping that we won't have to go through that again. We can't afford it. And so FaceBase, we hope, will save us.
Sorry, one more thing. So fixed share is more like a generalist repository. So do you sort of have any sort of annotations or standards or anything that you apply to your data before you share it?
Right. So there, no, we did not have much. I mean, well, we have some of the raw data with its metadata, but in general, you know, like the FAIR principles work, we didn't do a great job of that. And even so, it cost us almost $2,000.
So I have a question. Early on in development, these two glands, do they originate from the same primordial structure? And then do you look early on to see what differentiates them into these two glands? And it's, you know,
Right.
or is my my embryology old?
Yeah, right. So actually, that's a little bit controversial as to exactly, you know, whether there's contributions from no crest. Of course, there's no crest mesenchyme there, but in addition, other sources. And so we actually don't know. And why, let's say, very early, the two mesenchymes are different in terms of the expression. We're not sure if that's because of the different embryonic origin or not. And that's another question that could be explored.
And then there must be regenerative processes too that you can look at as well.
Yes.
Yeah.
So we're really impressed by the capacity of self-assembly in this system.
Yeah.
And it's and there have been claims that it may be possible to take stem cells, get them to self-associate. We'll just see.
You know, yeah. I'm thinking you could store stem cells from an individual.
Yeah.
And after they go under radiation treatment for cancers or whatever, to reestablish the gland potentially.
Yes, exactly. And I think one point we'd like to make is that it's very important to take into account the tissue environment. And so if you have a foreign, let's say, connective tissue mesenchyme, that's going to change the expression. So that's something to watch out for.